Welcome back for more physiology. Today we're going to talk about pressure, namely blood pressure. So we're going to deal with blood pressure and how measuring blood pressure tells us something about your circulation. We established earlier that blood flows from high pressure to low pressure, and that flow is described by Poissier's law. And we know that if we mess with any of the factors of that blood flow, like vessel diameter or blood composition or the total length of the blood vessels, the result will be flow will go down, probably, and your heart has to pay the price for it. If I were to take your heart and put it into the way that we have blood flowing and we actually start naming different parts, what we can end up getting is a very defined pathway of blood. In class, we talked about writing out a pathway going from right atrium through the tricuspid to the right ventricle, through the pulmonary valve, to the pulmonary artery, to the pulmonary lungs, to pulmonary veins, to the left atrium, through the mitral valve to the left ventricle, out the aortic valve to the aorta, taking some type of systemic series of arteries till we got to systemic vein or systemic capillaries, taking some series of systemic veins back until we get to either the superior or the inferior vena cava, which will then move back into the right atrium. That's what we drew out in class. I just don't have it written here. What we notice about blood flow, which has to go from high pressure to low pressure, is no matter what, the high pressure is going to be the left ventricle, because that's going to be the one that pushes the hardest, because it pushes through the systemic circuit of the body. But in order to create a circle that can keep going, that high pressure, the left ventricle, also must be the low pressure point, because otherwise blood would never flow back into the left ventricle. And if I can't get blood back into the left ventricle, I can't raise the pressure back up, so it can drive the cycle again. It's kind of like a roller coaster. You start with the highest point, and you go down until eventually you get to the lowest point, but you need something that connects the high point and the low point, the lift hill. When we look at blood pressure, we actually use a whole bunch of things to talk about blood pressure, and the most common one is we talk about your systemic and diastolic blood pressures. The term systemic means when your heart contracts, what's the highest point of pressure? Diastolic would be your heart relaxes, what's the highest pressure of a relaxed heart? We typically report the values of 120 millimeters of mercury over 80 millimeters of mercury, but it kind of depends on you. There's actually a range as to what is considered to be normal, and it really does depend on you. Whenever we measure these values, we care if you are calm and relaxed. So if you just did exercise or you were picking stuff up or you were going for a walk, these numbers are going to shift on you. So we want you perfectly calm, perfectly relaxed, not worrying about anything. Then we measure these values. The problem with measuring systolic and diastolic is that doesn't tell us the complete picture. So we have another thing that we can measure, which is the mean arterial pressure, and that value there, which we can calculate, it's not a difficult calculation, tells us about the condition of your arteries. So are they too elastic? Are they too rigid? And however the map changes, that tells us something about what's going on there. The reason why we use this only with arteries is because arteries actually have that diastolic and systolic pressures, systolic being the high, diastolic being the low. And as it fluctuates up and down, up and down, up and down, sometimes it's nice to have one reference point, hence the mean arterial pressure, and we can use that to judge what's going on with your arteries. Coincidentally, the up and down, the difference between the systolic and the diastolic is called a pulse pressure. And the reason for that is what the heart does is it makes the blood go from the diastolic pressure up to the systolic pressure, and then it drops back down, which is a pulse. So a pulse is actually not a rate of beats per minute. A real pulse is actually just a difference in pressure. Kind of like if you feel pulses of air at you, you feel little puffs of wind. If you had pulses of light, you see strobe light effects. A pulse inside of liquid, like blood, would be the exact same. But there's another value that we could turn out to measure, and that's called the mean systemic filling pressure. That value is actually a way of measuring what turns out to be the state of fullness of your body. So if you were to die and not have high blood pressure in your heart and inside of your arteries and low pressure everywhere else, and just let the blood pool everywhere, it's going to have a pressure. That pressure turns out to be what we call the mean systemic filling pressure. What your heart turns out to do is it takes that blood pressure from the parts that don't need it, like veins, and shoves it all into the heart and into arteries so that we can then have a cycle of blood flow. When we have a point where the mean systemic filling pressure is the pressure of the body, blood's not flowing in any way, shape, or form. The way that we measure blood pressure is we use a blood pressure cuff, which is referred to as a sphygmomanometer, and we listen for things that we call Korot cough sounds. So how do you do that? We can't measure the pressure of your heart easily. We can, but it's going to be invasive. So what we do instead is we measure the next closest thing that is easy, and that would be your arm. So what do? Put a sphygmo around your arm, pump up the pressure greater than that of the systolic blood pressure. When you do that, you cut off blood flow. We put a stethoscope in the artery just inferior or distal, more appropriately distal to where the cuff is. When we place the sphygmomanometer there using your fingers, not your thumb, what we'll end up doing is we let the air out of the sphygmomanometer, releasing the pressure. The moment blood starts flowing through the cuff again, what you get to do is you get to hear the heartbeat. You'll hear the pulse, the closing of valves. That sound will be the systolic blood pressure. We're going to keep doing that until eventually we reach a point that is below what the heart can push at, which is the diastolic pressure, because that's going to be its lowest point. What you'll notice is we'll get the heartbeat, bump, 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 bump. Then it stops. The reason why it stops is the pressure cuff is no longer pushing hard enough to make it so you hear the heartbeat. That up, down motion, the pulse, you're actually somewhere below it. And the result is you don't know that there's a pulse happening. So the sounds stop. The point where the sounds stop, we call that your diastolic blood pressure. So you're going to be doing a lab where you're going to be taking pulse and you're going to be taking blood pressure. In class, we talked about how you do this. The big deal, you don't push the sphygmomanometer up too high. 180 millimeters of mercury might be too high for a lot of people, so the 150 sounds better. You have to listen, so the louder it is, the harder it is to measure. When you measure blood pressure, you go off of what you hear, not what you see. And lastly, you never use your thumb. Just never, never, never use your thumb. I also gave a sheet of paper in class for students to read dealing with the placebo effect. And they were asked to answer these 5 questions dealing with the article in class, because we're going to be using this the next time we meet.